Good evening again. It is still Tuesday, July 7th, 6:52 p.m. For this video, we're going to look at the question of the possibility of this being a gain of the SARS-2 being a gain of function virus. So let's look and see what scientists and virologists have talked about when it comes to where this may have came from. So we'll start with an earlier report, and then we'll go towards a much newer one. So in a lab, altered virus, it's not natural. Like finding a dog with wings, there had to be lab tinkering. Take a bad coronavirus that is not infectious to humans, force its selection by culturing it with cells that express human ACE2 receptors, such cells having been created many years ago to culture SARS coronaviruses, and you can force the bat virus to adapt to infect human cells via mutations in its spike protein, increasing ability to bind to human ACE2, but reducing the bat ACE2 binding. Leading immunologists and geneticists have told Daily Telegraph there are two unusual aspects of SARS-2 that raise the possibility it was man-made rather than a naturally occurring virus. Number one, virus binds to human ACE2 receptor cells more strongly than it does to any other animal, including bats. Two, it has a furin cleavage site. Again, this furin cleavage site is key and also puzzles a lot of scientists as to how it got there. So it has a furin cleavage site that its closest genetic bat virus relative, RETG13, does not have. This site makes it significantly more infectious. Geneticist Dr. Ronen Shemesh, who's working on treatment for SARS-2, said in his opinion, the virus was more likely created in a laboratory than evolved naturally in nature. There are many reasons to believe that the COVID-19 generating SARS-CoV-2 was generated in a lab, most probably by methods of genetic engineering, he said. I believe that this is the only way an insertion like the furin protease cleavage site could have been introduced directly at the right place and become effective. Dr. Shemesh points to the insertion of the furin site as the most unusual aspect of SARS-2. I believe that the most important issue about the differences between all coronavirus types is the insertion of the furin protein cleavage site at the spike protein of SARS-2, he said. Such an insertion is very rare in evolution. The addition of such 4 amino acids alone in the course of only 20 years is very unlikely. And here's a more recent one that talks about the same topic. The virus is especially well suited and adjusted to attach themselves to cells in humans. Looking at human genetics, he pauses. So well, it's suspicious. First, this part of the virus is very stable. It mutates very little. That points to this virus as a fully developed, almost perfected virus for infecting humans. Second, this indicates that the structure of the virus cannot have evolved naturally. When we compare it to SARS, we see that there are altogether 6 inserts in this virus that don't exist in other known SARS viruses. 4 of these changes are suited to infect humans and could only be added most likely in a lab. Even if natural, the processes have most likely been accelerated in a lab. It is possible in nature, not likely. If the mutations had happened in nature, we would have most likely seen that the virus had attracted other properties through mutations, not just properties that help the virus attach itself to human cells. Explanation. Imagine that you have cultivated 1 billion coronaviruses you have gathered from nature. Then you take this massive viruses and inject them into a human cell culture. From, for example, the upper respiratory tract. As a result, a few of these viruses will change in order to better attach themselves to this type of cell in the nose and throat region, and therefore to infect humans more easily. You end up with a spike protein, which is perfect for attaching to and penetrating human cells. The properties in the virus we have yet to discover anywhere in nature. We know that these properties make the virus very infectious, so if it came from nature, there should also be many animals infected with this, but it still hasn't been traced to nature. Similar gain-of-function viruses exist, but only in labs, and there are cases where they have escaped. The logical explanation is it's from a lab. Those who disagree have the burden of proof. That's something to think about.